Okay, Chem 7, this is the biological molecules carbohydrates chapter. And so carbs play a super important role in our life because it provides the chemical energy for us to move and do all the work that we do as a human. And they're also formed by plants, so very important. And they're important for fuel, but they're also important for all of the molecules in your body. A lot of them have carbohydrate parts that are important on the cell walls for like flags to tell the body what things are, just signal the body what the cell is all about. So carbs are really important in a lot of different ways. So very complex and very unique. So we'll talk about simple carbohydrates, which are formed from sugars. And we've talked about sugar chemicals a little bit. And we talked about hemiketals, ketals, hemiacetals, and acetals. We talked about formation of those, and we'll see that in this chapter. So that stuff we talked about in the ketone and carbohydrate, carboxylic acid chapter comes back here in this chapter. So a lot of these sugars can be strung together in long strings, and when you do that, you use the word poly. So each particular sugar is a monomer, and when they're strung together, they make polymers. And so this is also in regular organic chemistry. You have strings of ethylene, that would be polyethylene, strings of propylene monomers into a long string together into a long unit is propylene. A lot of the fabrics we wear are polymers. They're strings together of small molecules all strung together in long strings. And polysaccharides, the starches that we eat in bread and complex carbohydrate foods like beans and oatmeal and things like that, those are long strings of sugars together and they form polysaccharides. So when we have one sugar, a saccharide is the same thing as sugar, and it's called the residue, just a fancy name meaning just one of these pieces, one of these sugars. One sugar is a monosaccharide. When you string them together, put 2 up to 10 saccharide sugar units together, it's called an oligosaccharide. And when you have more than 10, it's a polysaccharide. And some things just contain thousands of sugar units. So these monosaccharides, the one sugars, mono means one, saccharide means sugar. One sugar can be either formed from an aldehyde type of chemical, or it can be formed from a ketone type of chemical. So if it's if you see an aldehyde on it, it's called an aldo. If you see a ketone on it, it's called a keto. And then the second part of the name is saying how many carbons are in the sugar. So on the left side, I have 5 carbons, so it's called a pent. Ose means it's got a lot of OH groups on there, so it's a sugar. So ose is telling me it's a sugar. So aldo means aldehyde, pent means 5 carbons, ose means it's a sugar because it has all these hydroxys on there. So that's an aldopentose on the left. On the right, there's a ketone in that molecule. There's 4 carbons, so we say tech. And then ose, because it's got hydroxys on there, it's a sugar also. So it's a ketotetrose. It gets complicated, doesn't it? So another set of naming. We've learned about naming for alkanes, and now this is going to be a little bit different for polysaccharides and saccharides. So just another naming system that we have to learn. So ose means it's going to be it's going to be sugar. It's got a sugar in there. It's got a lot of those OH groups on there, hydroxys on there. And if it's an aldehyde functional group on the monosaccharide, the one sugar, it's an aldose. If it's got a ketone on there, it's called a ketose. And then the number of carbons in the chain of that sugar is named this way. So triose, tetrose, pentose, hexose, heptose, octose, nonose. And so you have to tell the person whether it's got an aldehyde in it or a ketone. You got to tell the number of carbons in there, and then ose means it's a sugar. So an aldotriose would mean it's got an aldehyde in it, it's got 3 carbons, and it's a sugar. It's got a lot of OH groups on there. An aldopentose would be aldehyde ending, it's got 5 carbons, and it's a sugar with a lot of hydroxys in it. Ketoheptose would be a ketone functional group on there. It's got 7 carbons, and it's a sugar also, ose. Okay, so those are monosaccharides, 1 sugar molecules. And the physical properties are they have a lot of hydroxy groups on there, so they're really, they've got a lot of solubility in water, and they have a lot of hydrogen bonds, so it's hard to melt them, change them from solid into a liquid. Okay. So those are the physical properties. So monosaccharides have chiral carbon atoms. We talked about that in the alkane section. When a carbon is hooked to 4 different groups, then it's called chiral. And glucose is the most important, I would say for you guys, a monosaccharide, because this is what we measure for blood sugar. And if it's too low, you're hypoglycemic in your blood. If you have too much in your blood, you're hyperglycemic. And so diabetics have to make sure that their glucose level is correct. So this is the molecule we're talking about when we talk about blood sugar, this D-glucose. It's got 4 different carbons here that are chiral because, for example, this carbon at the top has an aldehyde group hooked to it, it's got a hydroxy hooked to it, it's got a hydrogen hooked to it, and then it's got this big chunk down here. So this carbon, the top red circled one, is chiral. So glucose has 4. That means that these H's and OH's can be flipped around, and you're going to get isomers that are different. And so in our body, our body will accept D-glucose, but it won't accept another enantiomer, another isomer of this molecule. It won't fit correctly in our bodies. It won't be used right. Okay, and this is a little formula to find out how many isomers you have. It's 2 to the number of chiral carbons you have, or chiral atoms you have. So in this example, this D-glucose has 4 circle carbons that are chiral. So you put 2 to the 4th power, and you get 16 isomers that can form from this 4 carbons that are chiral in this molecule. So let's move on. All right, so we can draw these molecules with these little dashed and solid lines, and one is they're trying to show in 3 dimensions that 2 of these things that are hooked to the central carbon are pushed back, and 2 are going towards you. We're trying to show this in 3 dimensions, this Fisher projections up top here. And what I want to talk about on this slide is that when you have 2 enantiomers of these monosaccharides, it's because this OH group is either hooked onto the right side or onto the left side. And we use this little L and D to signify that. So L, just remember L means this hydroxy is on the left side, and D means it's on the right side. So these come from, I think, Latin words. So dextro means right side, and levo means left side. So I just remember L means left, And when I see this OH over here, that means it's an L isomer. And they're mirror images of each other. So we'll move on. And this is, remember, D-glucose is that one that we said is important in the body. And there's also one that's called L because these OHs are on the left side. So they're enantiomers of each other. And then if you can also change it into something called the diastereomer. But I don't really care so much about this. I just need you to know if it's a D or an L. So we're looking for that left or right hydroxy group. So important monosaccharides are 5-carbon ones and 6-carbon chains. And these ones here are pentoses, which means 5 carbons, and they're found in DNA and RNA. So these are the sugars that make up the backbone of the spiral for the helix for the DNA and RNA. So these are important sugars for that. And I said D-glucose is the one that we measure for blood sugar, and so that's the most important one in this whole chapter because I know you guys in health careers are going to be looking at glucose blood levels all the time for your patients. Galactose is another sugar. There's tons of sugars. Lactose is a sugar in milk, and then fructose is the sugar from that makes fruit sweet tasting. So there's lots of monosaccharides, mostly 5 and 6 carbon sugars are important monosaccharides. We also have ones that some of the OHs are replaced with H's, and they're called deoxy sugars. So you see like this one here, deoxy has H's only here. Okay. And some monosaccharides have added amine groups on there, amino groups on there. Instead of an OH, they have an NH2 group on there. And you've probably heard commercials on TV or, in your, on your phone or whatever ad you'll see for taking glucosamine to help you with joint pain and stuff like that. So that's another, I don't think they will work that well, but those are sugars that are replaced with an amino group, and that's called a amino sugar. Alcohol sugars are the ones that you see when you have sugar-free foods, sorbitol. They've changed it by adding hydrogens, reducing it, and they've changed it from a ketone or aldehyde. They've reduced it by adding hydrogens. Remember, reduction is adding hydrogens. So they've changed it from aldehyde into an alcohol. And this one here is another aldehyde to an alcohol, another aldehyde to an alcohol. So these are all showing how they're changing different aldehyde groups on the end there to something with just alcohol. So these sorbitol you'll see on sugar-free gum, xylitol. These are sugar-free things that your body cannot metabolize after it's been changed into these reduced forms. So they taste like sugar, but you can't use them for energy in your body. So they have no calories. We can also change a ketone aldehyde or alcohol into carboxylic acid. And these are called carboxylic acid sugars. And let me move on to see what else I want to talk to you guys about. Okay, so let's talk about how these little, I'm going to remind you that we can reduce aldehydes and ketones to alcohols. And we're going to remember to go back to that chapter where we talked about hemiacetals and acetals and ketals and hemiketals. Let me get to that part. So you can measure sugars in the lab by doing something called the Benedict's test. And we'll see that in a second. Let me find that for you guys. So this one's important when you're looking for glucose in urine, because you have a lot of sugar in your urine, and that's not a good thing. So this is a reaction that is done in the lab where they take copper to Cu2+. They do a reaction with the urine and to see if there's any blood, and if there is blood, it's going to have a color change. So it's going to change the Cu2+ into Cu+ ions, that sugar, and so that's going to show that you have urine, glucose in the urine. So that's not a good thing for your patients. Okay. So some sugars will show this positive test, some will not. So that's not super important for you to know, but just know that that Benedict's test is important for looking for sugar in the urine. All right, alcohols in these monosaccharides can form hemiacetals and hemiketals. So they form these sugars. We've been showing you them as flat molecules, linear molecules, but they actually at some time they're linear, and sometimes they're forming into rings. So sugars are very unique. So I'm just going to remind you that aldehyde plus an ethanol can turn into a hemiacetal. We're looking for this ROHHOR group. That's a hemiacetal. And a ketone in an alcohol can form into a hemiketal. We look for the RROROH group for a hemiketal. We learned about that back in the ketones and carboxylic acid section. Okay, and sugars can do the same thing. So let me show you that. Okay, so this is showing right here that this aldehyde, remember this aldehyde group here, is being catalyzed with the in a high acid solution. It can catalyze and form this ring. Okay, so we can form lots of lots of sugars are changing from linear structures to rings all the time. So they're going back and forth from linear to ring shape, back and forth, back and forth. So we can show it linear like this. And then it's also doing this reaction where it's taking this alcohol and reacting with the carbonyl carbon right there and forming a hemiacetal. So sometimes it's linear, and sometimes it forms into a ring, and sometimes it opens and closes, opens and closes. It's really complicated. But just know that sugars can be linear in form sometimes, and they can also form into rings. So for example, this little guy is opening, and then this carbon is flipping around, and the double bond O is flipping up to the top. So this is the open form of glucose, and you can either have the double bond O down, or it can have the double bond O up. And when it changes, it has an OH up, or it has the OH downwards. Okay, so and it's doing this all the time in blood solutions and water solutions and all that. It's going back and forth all the time. So when the OH is pointing up, it's called a beta. And when OH is pointing down, it's called an alpha. So this is alpha because it's down. This OH is down. It's beta because this OH is up. So I just wanted to show you that you're going to see that in the naming, and I just wanted to show you what alpha and beta meant. So the open form can switch back and forth. It can have the OH up, and then it can open up and then have the OH down. It goes back and forth all the time. It's in dynamic equilibrium, which means it's switching back and forth all the time. So there's different ring shapes for monosaccharides. Some is the Pectin form, which is this form that looks six-sided, and sometimes is the Urine form, depending on what it's made out of, and that's the five-sided ring right there. So you'll see both, depending on the molecules that are forming the monosaccharide, will decide whether it's going to be Pectin form or Urine form. Glucose is going to be that six-sided Pectin form, and then we have some that are going to be five-sided, which are the Urine form. Let's see. So once we talked about the sugars and how they form rings, those rings can then connect with other rings and form glycosidic bonds. So remember we talked about hemiacetal changing to acetal, hemiketal changing to ketal back in the carboxylic acid and ketone chapter. So that's happening in these sugars. So for example, maltose is the sugar that's in beer that gives it that is used by the yeast to form into the alcohol. So maltose comes from the malt that they use in the beer. And that sugar is two sugars, disaccharide. Di means two, saccharide means sugar. So two sugars hooked together makes that maltose that's used by the yeast to form into alcohol. And because this is downwards, it's called the alpha glycosidic bond. So it can also, some of these sugars can also do that copper test that we were talking about with the urine for glucose. And so this one here has an upwards here and downwards for this, so this is called beta. So there's different ways. I just want you to see that there's different ways that these can hook up. You don't have to memorize all this stuff. Just know that there's different ways that the sugar can form into disaccharides, and sometimes they're both going down, sometimes one's up, one down, sometimes they're both going up, and there are different ways of naming those. So it really gets complicated if you notice that these guys are hooking up in different ways. Here's another way that they can hook up. So it gets kind of crazy and complicated. So blood antigens are the ones like if you have A blood or B blood or AB blood, those things are called antigens, and these are the things that are stuck onto the cells of blood cells so that the body knows what type of cell it is. So like I said, sugars are like little flags on cells to tell the body more information about the cells. So this is an example of four little sugars hooked together forming an antigen flag on top of a blood cell. Homo Polysaccharides these are polysaccharides where you have 10 or more of these sugar units hooked together. Homo means they're all the same. So like a bunch of 10 or more glucose sugar rings hooked together. Hetero would be like a glucose and a maltose hooked together, or a fructose and a glucose, or something like that. They're different ones hooked together. So cellulose is all looks like this, and it's all these same kind of units hooked together. Very, very strong carbohydrate that gives structure to the plant cells. So cellulose is in paper. We can form plants into paper. Cellulose is a really nice, strong polymer made by plants, and it's tons of sugars going off forever and long, long, long lines of a polymer. And it makes a nice fibrous network that we use for paper and for other things. It's It's the structure of wood, and you know, the wood we make our houses out of. Very strong. And most little animals can't digest it except for like termites and things like that. So it's strong, pretty resistant to everything on the earth except for termites. We have other things, polysaccharides in nature, like if you've ever eaten jelly or jam, it's got pectin in it. And this is the structural network that's making the jelly, going from like grape jelly is made from grape juice, so we want it to be nice and stiff, so we use this network of sugars, this polysaccharide, to make it all thick into a jelly or jam form. All right, so in plants, it's pectin, which makes that nice jelly structure of jams and jellies, and it holds a lot of sugars in this beautiful network format. And this is really helpful to keep jams and jellies nice, and other like fruit lather and stuff like that we use pectin for, to make it that format. But we do the same thing in our bodies. We have glycogen in our muscles, and this is where we hold all the glucose in our body for when we need fast energy to do exercise and movement. We store glucose in the muscles, and then when we need it, a little enzyme comes in this network and pops off glucose units to use for energy. So we have enzymes that can get in this network and pop off the number of glucose molecules we need to do our exercise that we're doing. And so this form is really important because the enzymes can get in there and pop off the number of glucose needed to do the exercise that we have. If we had glucose just in a long line, it wouldn't be very helpful. This way, a bunch of enzymes can get in this network and pop off as many as we need as fast as we can get it. So it's very important that it's in this format so that our body can use it as fast as we need it. We also have polysaccharides in our that are lubricating fluids in our eyes and our joints. You might have heard of hyaluronic acid. That is helpful for the shock absorbing fluids that are in our bodies to help our joints work well. So that's another polysaccharide. It's hetero because it's got different sugars hooked together. Heteropolymer. You might have heard of chondroitin on TV for people who are having trouble with, their joints as well. So it's another polymer in our body. And that's it for sugars. I'm going to have one other lecture that you can listen to. It's similar. It's got a few other points in it. So I made two of them because you might like the format of one of the lectures over the other. So I have two lectures for this carbohydrate chapter. Thanks so much for listening. I hope that helped some to get you some information about carbohydrates. It's kind of complicated, so I don't want you to memorize everything. I just want you to know the basics of carbohydrates and how they're hooked together and why they're hooked together that way. Okay, talk to you later. Bye-bye.